Okay, this podcast is about probability in the Punnett squares. Probability is the likelihood that a particular event will occur. When you flip a coin, there's a half chance that they'll end up heads, the half chance that it'll end up tails. The same is true when you have, uh, or whether it's head or tails, it's random. So the way that alleles segregate during the gamete formation is also completely a random event. You see, gene, alleles are basically genes that are found on chromosomes. And chromosomes, when they segregate, so it's random. The principle of probability can be used to predict the outcomes of genetic crosses. So the question is asked for number on slide number 8. How is flipping a coin and similar to segregation during the gamete formation? Well, the fact of it is when when you flip a coin, it's random. When they line up on each side of the uh, meiotic plate, then it's random. Once again, the second question: how can the principle of probability be used to in relation to genetic crosses? It can show us how basically what the possible outcomes of a cross between and what the probability of having those outcomes. So Punnett squares are diagrams that illustrate the possible gene combinations that can happen during a genetic cross. To make a Punnett square for a one trait cross, you draw a box like the one over here to the right. So there are two parents, and each have two alleles. So you have four different possibilities. The two alleles for each trait are separated and placed into the boxes next to the boxes right here. So this is, for example, this one's big P, this one's little p. So we take the big P here, little p here. And it doesn't matter which one. You always have to, but you do have to distribute them through this. So you take one of the parts of the cross and put it on one side, and then one on the top. This represents the making of gametes. So when you're separating the alleles during the process of, it's also called meiosis. We'll talk about this later on. So, whoops. So the question is asked, what are Punnett squares? They are a diagram to help to illustrate the possible gene combinations and their possible outcomes from a cross. So uh, what are they used for? They're to help us to be able to predict what might happen in a cross. So an allele from each parent is combined in the boxes. So the big B or the big P here is combined with the other big P from up here, and the combining of the alleles represents the fusing of the gametes during sexual reproduction. So Punnett squares make interpretation of the crosses much easier. So what you need to do is we are going to make make a Punnett squares for the following crosses. So you're going to make a big B, big B. So it looks kind of like, let's just go ahead and draw one real quick here. And I'm going to have you do one and I'll do the other. Whoops, hello. So here's I'm going to go ahead and do the big B, big B. So it doesn't matter which one you do, where you put it. I'm going to put the big B's on the side because I always put the first ones right side. And then on the top, I'm going to put the little B's. Then that is the segregation. Now we're going to put them together. Big B, little b. Okay, big B, little b. This little b is here. This little b is here. These big B's are here. Okay, that's kind of the way it works. Okay, so you're going to do the same thing for the big A, little a, and big A, little a. Once again, you're going to put one of the big A's down on here, and the little a here, and so forth. Okay, so that's that. So what does combining of alleles in the Punnett square represent? It represents the sexual reproduction. What happens when the gametes come together in sexual reproduction? So the types of gametes produced by each of the F1 parents are shown along the top and left sides. So that's this. The possible combinations of the F2 offspring appear in the four boxes down below and make up the square. The letters in the Punnett square represent the alleles. So these are the alleles. So number 11, what do the letters along the top and the left sides? Those are the gametes. What do the allele combinations in the boxes? Those are the possible offspring. And then the Punnett square to the right shows three different allele combinations, big B, big B, big B, little b, and little b, but only two different physical characteristics, purple flowers and white flowers. So here we see these, notice that these big B, those are purple too, because this is the dominant trait, the purple is. Notice that the recessive one is little b, little b. To distinguish between the different allele combinations in the organisms with the same type of alleles are called homozygous dominant. So if we have two big alleles, then that's homozygous. If we have two little alleles, then it's homozygous recessive. So the question number 12 for slide number 3 on what does homozygous mean? It means the two of the same alleles. Homo means the same, zygous means alleles. So when both alleles are dominant, they are called homozygous dominant. And when they are recessive, we call it homozygous recessive. So when both alleles are different, then we call that heterozygous. Hetero means different, and then zygous means alleles. Homo, same, zygous, alleles. So homozygous, same alleles. It could either be little b, or big B, big B. So the question is asked, what does heterozygous mean? It means that they're different alleles. The plant's physical traits, purple or white, is called the phenotype. Pheno means physical type. The plant's allele combinations would be the genotype. Geno stands for genes. Comparisons can be made between the different phenotypes, like 3 to 1, 3 to 1 of the white, purple to white. And ratios can also be made for genotypes like 1 to 2 to 1. You always have to put the whatever with the genotype. You can't just say 3 to 1. You have to say 3 to 1 purple to white, or 1 to 1, 1 to 2 to 1, big B, big B, to little b, little b. And it's easier to write it than it is to say that, especially when you have so many b's. Anyway, so the question is, what is a phenotype ratio? It's a comparison between the different phenotypes for a trait. What is the genotype? A genotype is a ratio is the comparison of the gene types of genes. Makes it easy to remember. Okay, so when we do a test cross, Mendel devised a system of conducting verification for the results obtained by him. So remember when he made sure his purebreds were purebred? This is how he did it. The test crosses involve a breeding an organism that is dominant phenotype. You can't tell whether it's either the heterozygous or the homozygous recessive. And you put it with a homozygous recessive individual. Why? Because the homozygous recessive individual will, if there is one recessive allele, then the offspring will end up having the recessive trait. Well, at least some of them will. Because if all offspring display the dominant phenotype, the individual in question is the homozygous dominant. Okay, so if we were to take and put, for example, big A with little a, then all the individuals would be the dominant trait. If the offspring display both the dominant and recessive phenotypes, then the individual is heterozygous. So that's what we're talking about when this shows here. So the question is asked, what is a test cross? It's to make sure that a individual is a homozygous dominant or purebred. And then what is it used for? To make sure that they're dominant trait. So 6, how does a test cross work? You cross the unknown individual with a homozygous recessive individual. And then how does it work? If any of the offspring show up with the homozygous recessive trait, then it means that your individual was heterozygous. If they all ended up with the dominant trait, then you have then it is a purebred animal. So a F1 plant that is homozygous for shortness is crossed with the heterozygous F1 plant. What is the probability that a seed from the cross will produce a tall plant? And draw the Punnett squares for this crosses. So how do we do this? Essentially, what we do is, let's just go ahead. We'll just move this right here, and we'll make a cross. And so, hello, this is going to be hard to do. So heterozygous individual, big T, big little t, and big T, little t. And you're just going to go ahead and fill in that. Okay, so